ZombieTech. Welcome to ZombieTech, a forum for engineers, scientists, and inventors to ponder on the technologies needed to survive the inevitable zombie apocalypse.
She's Addie.
He's Whisker.
And who do we have this week?
This week we have Tara Smith, an associate professor of epidemiology at the University of Iowa, as well as ridiculously cool, science blogger on science blogs, and part of the Zombie Research Society.
Now, is that the same thing that ZombieTech is part of?
Yes. So
because he was fun to talk to.
Yes. So he's the crazy MRI neuroscientist that we talked to way back when. Yeah. And now we have a chance to talk to the expert epidemiologist of, freaking Zombie Research Society. Hi, Tara. How are you?
Hello. Thanks for the invitation.
Anytime. So, I was mentioning to you 2 seconds ago, epidemiology is this crazily, intimidating concept. You know, we hear about it all the time when some crazy virus is on the move, but we don't really understand everything that goes around it. So I was just wondering if you would be willing to tell us a little bit about what epidemiology is and how you got started into it.
Sure. Epidemiology first is a huge field. So at its core, it's just looking at how diseases develop and spread in populations. So, why do some people get lung cancer? Why do some people die of lung cancer and others can, survive? Obviously, my field is infectious diseases. So, what we look at specifically is a bacterium called Staph aureus. And we look at how that's transmitted within populations, especially in rural areas, and also how that spreads between animals and people. Mm-hmm. So a lot of epidemiology involves, looking at disease patterns, looking at who gets diseases, who gets severe diseases, and, why some people seem to be protected. So that at the core of it, it's really about trying to prevent disease by understanding it.
Sure. And then how did you get into it?
I was always kind of an infectious disease nerd. When I was, in the high school era was when there were a lot of the Ebola outbreaks, and Laurie Garrett's The Coming Plague came out, as well as The Hot Zone. So both of those were kind of big books when I was in high school. So, I was interested in those, and I've always had an interest in disease development. My mother has multiple sclerosis, so that was a big impact on my childhood. Was why did she get this? And millions of people are healthy, and is that a risk for me as I grow up, and things like that. So I started thinking about these things probably at a much younger age than most people who go into epidemiology.
So that whole memo of you're supposed to be into dolls and beating and scrapbooking just got lost in the mail somewhere?
Yeah, not so much. I was actually, one of the things I really liked as a child were detective stories. So I read a lot of Encyclopedia Brown and Nancy Drew and things like that. So, that still kind of, I think, played a role into my development as a scientist because I always wanted to find things out.
Sure. So what kind of training does it take to become, say, an epidemiologist who works with Staph aureus?
Uh-huh. And again, it's a broad field. So people come into this from all kinds of different backgrounds. I was a biology major in college, and initially, again, kind of in high school, I was thinking maybe I would do medicine, but I did some rotations in the hospital and didn't really like kind of the one-on-one medicine thing.
Sure.
So I found public health, and that was a way to still do things in the medical field, but to do it from a population perspective, and ideally prevent disease in a lot of people instead of just kind of one at a time. So I did my BS in biology, and then I did a PhD in microbiology, and then postdoctoral work in infectious disease epidemiology.
Wicked awesome. Yes, actually, I do work with infectious diseases, so that's great. So, in terms of the research, I mean, obviously, there's a lot of number crunching. From my understanding of epidemiology, the public health specialist will be like, oh, okay, well, this office or this hospital has called in, 650 cases during this time span or whatever, and this hospital here, about 10 or I don't know, 50 miles away has come up with 300 cases, et cetera, over this time frame. So I'm just gonna number crunch. Now, since you've had experience in like microbiology, which I imagine is a lot of lab work, and like more wetware, I guess, in terms, as opposed to software. Like, are you still able to do that type of, like swabbing cheeks or I don't know,
Oh yeah,
taking
yeah.
blood samples and
Yep, I still have a large wet lab that is actually the basis of most of my work. And since we do things in both people and animals, some weeks we just finished up a big cohort study where we were in 33 different counties in Iowa over one summer, enrolled about 1,300 people. So took swabs of their
Wow.
noses and their throats for Staph aureus. So we're wrapping that up and then we come back to the lab with all of those samples, isolate the bacteria, then do all the molecular work on them as well. So we have that, and then we also, since we do animal stuff, we'll be swabbing, in people's houses. We'll swab their dogs and cats and guinea pigs. We go on pig farms, cattle farms, take nose samples, sometimes take samples from the other end, which is a little less
Wow.
pleasant for the students.
Wow,
But
sure.
so we do a lot of kind of primary sampling as well as, data analysis.
That is super freaking cool.
Yeah, it's a lot of fun.
I can see where the animal thing would be kind of important too. We were on our way back from California recently. We were driving and we went through northern Texas and we went past some of the factory farms up there, and the monoculture of millions of genetically almost identical animals packed
Right next to
butt
each
to
other.
nose
Mhm,
as
yeah.
far as the eye can see, that it presents a huge wonderfully resplendent environment for little bugs to replicate and
Absolutely.
mutate.
Yeah, and Iowa is #1 in pig production and #1 in egg production, and about 7 in turkeys and 8 or so in cattle. So we have,
Wow.
I mean, almost an order of magnitude more animals in the state than we do people.
That's
So
awesome.
it's pretty crowded as far as some of the animals go.
Sure. So, okay. I think when I was reading up some of like the things you've written, there was an interesting article about where you guys got samples from, I don't know, it was like 13 different stores of their, I'm guessing ground pork or pork chops or what have you. And you guys were sampling for Staph aureus, whether it was methicillin sensitive or methicillin resistant.
Uh-huh.
And for those who don't know, methicillin resistant Staph aureus MRSA, that in my field, because I'm a nurse, and MRSA blows. And I don't mean that just because it's like sticks around in the nose, but it blows because it impedes healing. It keeps people in isolation. So I have to gown up every single time I need to go and just say hi to the patient, what have you. So it's a very big inconvenience, and it costs patients a lot of money.
Yeah, it's a mess.
Yeah, it truly is. And so you're looking for MSSA and MRSA, and I think you found MRSA in some of the pork products.
Yeah, we did 2 studies, and the second one was the biggest one. And in that one, we found MRSA in about 7% of the pork products. And it didn't matter. What was interesting about that one was that it didn't matter if they were kind of your factory farm, pork, or organic pork, which we had expected, to show lower levels.
Sure. Huh. So what's the likelihood that a human could get? So say the pork is improperly cooked, right? What's the chance that, do I have a 7% chance of getting MRSA?
Well, even if it's improperly cooked, I mean, staph can cause food-borne disease, but usually when it does that, it's actually due to a toxin it produces. So you get staph food poisoning more from like, your Aunt Betty's potato salad or something that's been prepared. Maybe she had staph on her hands and then it kind of sat in the fridge or at room temperature for several hours. The bacteria produced the toxin. Yeah, and then you get it that way. So from the food poisoning aspect, we don't think that's probably a big deal. And for that, because it's toxin based, it doesn't matter if it's MRSA or if it's methicillin sensitive, because you're not going to treat it with antibiotics anyway. You're just going to kind of wait it out. But what we're more concerned about is when you handle the pork products, or we found it on other meat products as well. So people don't always use the best food handling procedures.
Sure, yeah.
So if they touch a pork chop and then they, rub their nose or something, or don't wash their hands or don't wash their counters afterwards, then Staph aureus sits pretty hardy in the environment. And so it could stay there potentially for, hours or some cases days, or it could, live in your nose then for weeks or months. So that's more of our concern, is that people are handling it and not, handling it very carefully. And
I have a question for you.
Sure.
Now, is this the sort of thing where if you're exposed to it, your immune system builds up more of an ability to defend against it and keep it out of your system? Or is it just sort of...
Unfortunately, with staph, not so much. There have been some studies looking at people who are carriers. So, as was mentioned, most people who carry this, it's in their nose. Sometimes it's in their throat. It can also be in your skin or in your gut. But people can carry this for months or, in some cases even years, that they've tracked people and they're carrying the exact same strain for, years at a time. So we don't really understand how the body responds to it, but we don't seem to have a really great immune response that, kind of knocks it out.
And is this because it's, on some people, it just doesn't affect them adversely enough that the evolutionary, process has made it a priority? What do you think?
Yeah, that could be part of it. I mean, it in most people, it doesn't cause disease. Most people carry it in their nose. They'll never get sick from it. So they'll never really have that, active immune response, like if it was in your blood or something like that. So yeah, it's just, it's kind of co-evolved with us probably for, millions of years perhaps. And we just seem to get along with it pretty well until it breaches one of our defenses and wreaks all kinds of havoc.
And that's exactly usually what we see in our patients too, is that, they've probably been carriers for years and years, but but it's not until they get heart surgery before it shows up its, ugly head.
Right.
And just causes, infections.
Yeah, if you have any kind of an open wound or something, I mean, it's just, that's when it goes crazy.
It's like, hey, it's a party now.
Yeah.
Yep. So then, okay, so this study comes out, right? And I'm like, oh my gosh, MRSA and pork, because I love cooking with pork. I mean, sausage, like I'm Chinese, so we use pork all the time. I'm like, what is this? Not like, oh my gosh. But I'm relatively educated when it comes to this type of thing. So, when, say, the media gets a hold of this, right? And is like, oh my gosh, community acquired MRSA is found in pork. For those who would like to think that we're more educated about like discerning what is hype and what is reality, what should we look for in, like, say, these media reports or what have you?
Meatball recipes, I think.
Yeah.
About
what's important.
I would say that anything that is tempered with some commentary by a microbiologist or somebody who understands this. I mean, meat is not sterile. Nothing we have in our home is going to be sterile.
Right.
So, there's not a need to panic about some of this, but it is just, emphasizing again, safe handling processes, safe cooking processes. Just be careful and realize that there are going to be all kinds of bacteria, all over not only your meat, but anything else you touch. And especially if you're, immunocompromised, if you're sick in some way, if you have young kids, if you have elderly people in your family, they're going to be the ones who get hit harder with this. So just be careful and use normal cautions. I mean, I don't go around following my kids with a bottle of hand sanitizer after everything they touch or, I mean, I'm not a germaphobe. I know that there are many things out there that can harm us, but there are orders of magnitude more that
you're still healthy
or help us. So,
you're still healthy after all of the handling anyway.
Exactly.
So I'm gonna transition then from going from
Well,
MRSA.
hold on just for a second. So
Oh,
now we've got an expert here in this topic, and it does come up quite often. So I thought I'd ask while we had.
Whiskey's very fascinated by
Yes.
like antibiotics and stuff. Yeah.
Yeah.
Go for
Because
it.
it seems to me like our immune system is basically the more stuff it bumps up against, typically the stronger it gets. I mean, if it has ways to deal with creating antibodies for the stuff anyway. So it seems to me like using or overusing antibiotics and sanitizers and stuff like that, especially around kids, is probably a really, really bad idea because their systems don't build up the kind of immunities that, say, I built up playing outside as a kid. And then they get sick more often as adults. And I think that's kind of sad that by trying to keep them safe, you make them more sickly as adults. It's kind of, I don't know. You're the expert. Is this the case?
Yeah, well, what you're talking about is basically been formulated into what's called the hygiene hypothesis. So it suggests that the more you are exposed to some of these organisms, especially as a child, that the healthier you tend to be as an adult. There have been some studies that support this pretty well, others that are a little more conflicting, but there's definitely been a body of research out in the last 10 years or so that does suggest that, the more times you get antibiotics as a kid, the more likely you are to develop things like allergies and asthma, basically where your immune system is overreacting to all of these little things that, maybe had you not had as many courses of antibiotics or something when you were very little, that it would have learned to tolerate over the years. So there is some research out there suggesting that you definitely don't want to overuse antibiotics and you don't want to kind of overprotect your kids and try to keep them in a bubble. But I mean, as a parent, it's a very hard balance to strike, right? Because, there are certainly bad germs out there that can do your kids harm. And
there's a lot more than just germs out there, too. I mean, if we're talking about something like a parasite, or there's other things that your immune system just, I mean, there's nothing that it can really do to learn how to fight those things, and you definitely have to avoid them, and cooking your meat properly helps with that. I don't know, from personal experience, I never had any antibiotics as a kid or an adult, and you know what, I don't get sick as an adult. I just don't. Everyone else does, I don't, and maybe that has something to do with it.
Yeah, and my son, so I have two kids. I have a 13-year-old daughter and a 10-year-old son, and my son had chronic ear infections when he was a kid, and so was on antibiotics many times, and he has asthma. And so my daughter, never had any of those issues, and is perfectly healthy now. So I mean, just anecdotally, and of course, the plural of anecdotes is not data, but just from my own personal experience, I've seen that in my, my own family.
Sure.
Yeah, I'm just sort of slowly collecting these anecdotes as we go along.
But she's just like, see, I told you so.
We get to talk to a lot of people who know enough about this kind of thing to make objective observations on these anecdotes. So I think it's useful to bring up the conversation when it's appropriate. Anyway, you were saying, Eddie?
Right. Sorry. So before we were talking about like MRSA and its spread, but we are ZombieTech, and ZombieTech is, I mean, an epidemic of epidemic proportions.
As much as we eat pork, we're probably
proportions.
staff zombies, evidently.
That would be awkward.
Yeah.
Because we'd be the first.
I don't know, my nose itches now. I'm not quite sure.
So then when we start hearing, say, on the media about an epidemic or a virus and maybe some sort of announcement from the CDC, again, how do we differentiate between fact and media hype, besides, of course, like seeing the official CDC reports or what have you?
I mean, in general, it's difficult because, of course, this summer there was all of the reports out about the zombie bath salts and all of these zombie attacks everywhere, and everyone went nuts.
Which were actually from marijuana.
Yeah. In some cases, yeah. I mean, it was, but it just all went back to, is there this big zombie outbreak, and
Of course.
I mean, every case where someone bit someone, it kind of came up on all the zombie blogs and stuff. So
I think all of our listeners are
was an excellent
probably
week.
very proud of us that we steered mostly away from all those
Oh.
topics.
I was
Yeah.
well.
yeah, but I mean that was definitely a lot of hype, and it just got so
It was fun
out
to
of
watch. Yeah.
control. Yeah. So you have that kind of that extreme
Sure.
on one end.
Sure.
But I mean, I think there are some really good reporters out there who cover infectious diseases that, if they said there was some kind of a zombie outbreak, I would trust them implicitly to know what they're talking about.
Gotcha. Any names? Let's name drop.
Yeah, so Maren McKenna actually is an infectious disease writer. She wrote a book a few years ago about the CDC, the Epidemic Intelligence Service.
Okay.
So kind of the disease detectives at the CDC. And then more recently, she's written one actually on MRSA called Superbug. So she actually also writes a blog called Superbug that keeps up to date on all of these infectious disease topics. So she's very good with all of this and is very tuned in and has great contacts in the scientific field. Helen Branswell is another one, and she's out of Canada. And she's covered a lot of flu research, and she also was there for the SARS outbreak in Canada in
Sure.
2002 or whatever. So she's covered this beat for a long time. There's also a website called CIDRAP, which I'm probably going to forget the acronym now. It stands for the Center for Infectious Disease Research and Policy, and that's out of the University of Minnesota. And that's basically their whole beat is covering infectious disease. So some of those I would totally trust if they told me that a zombie pandemic was happening.
Mm-hmm.
There's also a reporting service that I subscribe to called ProMed Email.
Okay.
And they keep track of epidemics all over the world. So this is something, on that it would probably start out as like an inquiry about, some weird zombie-like syndrome or something, and then kind of morph as they got more information on it and more scientific confirmation and things. But that's another good site for outbreaks of anything all over the world.
Sure. So, what constitutes as a pandemic virus?
So if it is pandemic, that means that it's gone basically all the way around the world.
Okay.
And there are, the WHO has some very specific definitions of what constitutes a pandemic, and it has to be kind of widespread outbreaks in, I forget exactly what it is, but some number of WHO regions.
Mm-hmm.
So then, if that's happening, if it's some widespread virus in the U.S., in Africa, in Asia, in Europe, that would probably be enough to declare it a pandemic virus.
So, like, have you seen I'm sure you've seen the movie Contagion.
I have, yep.
Yeah, so that would have been like uber pandemic.
Absolutely. Or even recently here, the 2009 H1N1 influenza.
Yeah.
We watched that unfold in the period really of about a month.
Yeah.
Where it started out kind of these reports of localized infections in Mexico and then in California and then in, U.S. tourists coming back from Mexico and then New York school kids that had never been to Mexico. And, it just went from this very localized and isolated infection to, being completely around the world in a matter of maybe 6 weeks.
And how much concern with that? Because I personally, I was kind of like, oh, it's media hype. Just, wash your hands.
Uh-huh.
You'll be fine. But, I mean, there were people who were like foaming at the mouth for for vaccines.
Uh-huh.
So like, what what should I actually have done?
Well, no, I mean, that's the scary thing is that, initially when the first reports came out, it was that, these young, healthy people had died from this flu, which, I mean, flu is it's a big killer in the U.S. It kills about 36,000 every year.
Mm-hmm.
But most of those are, people who are not young and healthy. They're mostly the elderly, the very
Right.
young. So the people who are more likely to die from any,
Anything.
event.
Yeah.
Yeah.
Mm-hmm.
So it was concerning, to us kind of in the infectious disease area that it was killing apparently, young and healthy people because that's what we had seen in the 1918 flu pandemic
Mm-hmm.
was that, the people that it hit the hardest were people from about age 25 to age 45 who hardly ever die from a normal flu.
Right.
So initially, we were concerned about that, but then it turned out, as more and more cases developed, that this was actually a pretty mild flu. It still hit some risk groups hard. Again, my son has asthma. That's always one of the big risk groups.
Mm-hmm.
So, some children with pre-existing conditions like asthma, did die from it. But overall, it was pretty mild. But, it took us really many months of good surveillance to be able to say, yes, this is flu, it's pandemic, but the death rates are, equal or even lower than a normal yearly flu.
Mm-hmm. Mm-hmm.
So it takes a lot of, good, careful research and surveillance to be able to say that. And in the beginning, we just didn't know.
So was it erring on caution then?
Yeah, I mean, with public health, there's what's called the precautionary principle, which is kind of like, for an MD is first do no harm.
Mm-hmm.
So it's kind of like that, but on a population level. So that, we wanted to protect people. We wanted to, get a vaccine going. We wanted to make people, aware to wash their hands,
Mm-hmm.
Try not to transmit it, stay home if you're sick. There are some drugs that can help minimize flu. It's not really a cure, but they
Just
can
to
lessen
ease
the
the
severity of
symptoms.
symptoms.
Sure.
Exactly. So,
Mhm.
Have people, especially who are in those risk groups, know that those drugs are available. You should take them very early in the disease, all this stuff that got out there. But I think that those messages were kind of overwhelmed by the, oh my God, we're all going to die type of stories.
Yeah. Yeah. Okay.
Yeah.
Got it. Because I was willing to wait until, some conclusive evidence about its actual severity
Uh-huh.
was before I went out and got myself a shot that had been developed in the course of like a couple months.
Yeah.
Yeah. So
There's been problems with rushed stuff like that in the past where they've come up with these vaccines and the vaccines have caused more problems than they've fixed.
Well, and actually, so that's another question. I
it's
think
not the kind
it
of
was
thing
in
you
the
want to rush
70s.
through.
Yeah, like in the 70s, there was also swine flu, H1N1, and some of our friends, they sent us these YouTube videos of like swine flu propaganda, which I'm sure you've seen. About how like, oh, if you don't get it, you'll end up like this dead-looking person.
Right.
And I think in that, was it the vaccine ended up causing a number of cases of, I think it's Guillain-Barré syndrome.
Yeah. Mm-hmm. Well, so they've done some retrospective analyses of those data, and looking at Guillain-Barré in flu and flu vaccines in general, and actually with the 1976 swine flu vaccine, it actually did not cause, I can't remember the exact data, but I mean, it wasn't a significantly increased incidence of Guillain-Barré over what you would expect from a regular flu vaccine year. And many of the recent papers out have looked at people who get Guillain-Barré who were, unvaccinated versus vaccinated. And they've actually seen that getting influenza infection is a much higher risk of developing Guillain-Barré syndrome than getting the influenza vaccination. So getting active flu infections actually make it much more likely that you'll get that disorder rather than getting vaccinated flu. So yeah, it's actually quite protective to get your flu shot.
Sure. So it's actually interesting that you mentioned that, because I think I also read something that said that those who ended up getting the vaccine back in the 1970s were actually had a significantly higher resistance to the more recent H1N1, which would, make sense if it's essentially, a similar strain.
Right.
So
I don't know, man. Everybody that I know that gets the flu vaccine gets the flu every so often, and I don't get it.
As a healthcare provider, I am required to say that is a myth and a what's the word? Like a urban legend.
Yeah.
Mm.
Yeah, the flu vaccine is a killed vaccine, so you're not going to get the actual
Oh, no, I'm not saying
flu
you get
from
it from there. I'm just
Oh,
saying
okay.
that there's enough strains of it that if you get
Oh, sure.
vaccinated for one of the strains this year, that doesn't mean it's going to be the one that comes through your neighborhood.
Well, but there's actually multiple strains in the flu vaccine. And it's tricky because lots of people will say, well, I got vaccinated for the flu, but then I got the flu.
Mm-hmm.
But there are so many flu-like illnesses,
Mm-hmm.
and lots of people don't even realize what actually influenza is. They'll get a diarrheal disease and say, I got the flu, because they're thinking stomach flu.
Sure.
But that's totally not what the influenza virus, does. It's a respiratory disease, muscle aches, fatigue. So, unless you actually get tested at your physician, and they say, yes, you have influenza virus, then, you can't be 100% sure that you had your flu shot and then got flu, because you might have had
Could have been any number of things.
rhinovirus, adenovirus, any kind of,
Mhm.
there are hundreds of respiratory viruses that you could get.
Yeah, last time on ZombieTech, we were talking to another bio person, and she was sort of going off on how much more we've been learning lately about the actual flora and fauna culture inside of the human body, thanks to the ability to do deep sequencing on all of the little critters running around inside of us.
Uh-huh.
And man, oh man, there is so many things that could cause those kinds of symptoms inside of a human body.
Mm-hmm. C. diff. Tasty.
Oh, fun.
So, it was interesting. In terms of like swine flu, I remember they were trying to institute quite a number of quarantine areas, in the airports or what have you. Now, in movies, nobody ever respects the quarantine.
Uh-huh.
Like the protagonist has to get out and save their family or what have you, only to shoot them later because, So let's talk quarantine. How effective is this? And how little do people
Against
actually
what? Zombies? Terrible. I've seen enough movies to know it's terrible at being effective.
Yeah, it just ends up everybody being zombies.
They're all in the same spot, and then they're zombies, and then they're all big group of zombies, and that's just bad.
So let's talk quarantine for like something a bit more normal, like the swine flu, and then let's talk quarantine for the zombies. How about that? Yeah. So how effective is it actually?
All right. So quarantine, when it's done well, it's pretty effective. So just one, kind of clarification. So people often to group kind of quarantine and isolation in together, and they're not exactly the same thing. So if you're been exposed to, a potential infectious agent, but you're not necessarily, so it's if you're around someone who's had H1N1 or something like that, but you don't necessarily, have it yourself yet. Isolation is when you are sick and they put you in, kind of your own little room, away from everybody else. So those two kind of get mixed up sometimes.
Sure.
So quarantine is the one that is more effective because, again, you're taking a person within the whole incubation period of disease and putting them away from, kind of society at large. The problem is that, of course, that's really tough to do because, you do get resistance from people saying,
I have my
I'm
human
healthy.
rights.
Yeah. Well, yeah. I mean, healthy. I'm not sick. Why are you putting, why are you doing this to me?
Right.
You don't have the right to do that. You're taking me away from my job, family, what have you. And it, totally understandable from the individual level. But, it becomes an issue when you're protecting the population at large, and actually governments, do have that power to put you away. And they can, if you don't obey that, they can put you in a, an isolation room in jail as a kind of a last resort.
Sure.
So, if it's done well and people adhere to it, it can be very effective. And that's actually quarantine and isolation were really two of the things that helped stop the SARS outbreak. Because, especially, Asia already, many countries in Asia already are better at doing this than us. Their populations tend to be a little more used to it, a little more compliant. They're the ones that you see anyway. Anytime there's any kind of outbreak, people are voluntarily using masks, things like that.
Right. Even when they're not sick. Yeah.
Right. Exactly. So they're, I think, a little more attuned to things. So that helps kind of stem it in, Hong Kong and some of the other areas where SARS hit. And then they also use that in Canada and looking at, especially when this broke out in hospital settings where they would, isolate patients who had disease and quarantine, not only family people, but in some cases, healthcare providers who were exposed to those patients. So, that was one of the things that helped break the chain of transmission for that and kind of get that under control.
I think here it's almost impossible to, behave correctly in this kind of a situation because we put vanity above science. Nobody's going to walk around wearing a mask. They would be seen as a hypochondriac. There would be serious social consequences for doing that in our country because we're so ignorant.
Well, and masks are, especially for the general population, they're not the best thing. In some cases they can help, but, people wearing masks, especially if they don't know how to use them correctly, they're not maybe a healthcare professional or something, they'll pull the mask down to talk to people. It completely defeats the purpose. Or they'll take it out and smoke or, so we've done some studies on, farming exposures and, trying to protect workers from, the nasty viruses and bacteria that might be on these farms. And masks are one way to do that, but they're just never used correctly, or they're reused so many times that they're just completely useless. So, so it's kind of a a suboptimal intervention anyway.
Sure. Now, so that's like SARS or quarantine in terms of SARS and H1N1. What about zombies? What kind? If you were like the director of zombie epidemiology, how would you institute quarantine?
Yeah, and that's it's difficult because so one thing, we would want to know everything we could about this, new zombie virus, bacteria, what have you. Because one of the hardest things is when you have this any kind of new infectious agent is you don't know very much about the incubation period. Because with a quarantine, you usually put people in in their quarantine for usually at least 2 incubation periods of the pathogen.
Okay.
So if the incubation period is, well, you have something like 28 days later where it's instantaneous, then, you'd only have to have someone quarantined for like a day because if they were exposed, they'd either turn right away or, or not.
Right.
But, if you have some of the longer acting zombie viruses, something like more like The Walking Dead or some of these other kind of slower viruses, you might have to quarantine them for maybe a week, maybe more. So that's the first thing that you'd really need to know. And, that's difficult without having good studies of the epidemiology of these viruses. And in a zombie outbreak, that's going to be tough to do. To do.
Well, let's say it's the 15-minute. Let's say it's a 15-minute period between, bite and conversion.
Mm-hmm. Yeah, then quarantine isn't going to do very
anything.
Much. You're not going to be able to reach those people in time. And you can see that's with, like the 28 weeks later, because then they added in the carrier state.
Yep.
That they had these people who were, bitten, infected, but weren't all zombified yet.
Right.
But they could pass it on to other people. So that's the other thing is that, for people who may have been exposed, but are still looking healthy, still not showing symptoms, do you just proactively quarantine them all?
And if the virus is a successful one, it would probably be closer to that.
Mm-hmm.
Yeah.
Yeah, that's, we see carrier states with almost everybody. There are people who are, infected even with, flu or something that are asymptomatic. So they're not showing any symptoms, but they can spread the virus, happily. You see that with kids with flu. They can be asymptomatic and shedding virus for 21 days. These little germ factories, and you don't know it. So, yeah.
Huh. Yeah, that's totally me, I think. My immune system is awesome. I never get sick. Want to shake hands?
You spread it to everyone.
Yeah. It's awesome.
You're the Typhoid Mary, huh?
Now, my approach to zombie quarantine is more along the lines of travel around the country, kidnap everyone like Tara, get them as far away from other people as possible so that they don't try to be heroes and save the day and get turned into zombies because we need her brain afterwards to rebuild society.
Yeah. But I wouldn't see, I know this very specific niche, but, You also need some of the MDs and things like that too.
Yeah,
Oh,
we
have
have
that.
yeah.
Yeah, we have.
This is episode 59.
Yeah.
Oh, yeah.
We've
yeah,
got a
so you're
list
good.
of 50 some people that are going into
Into your bunker.
Yes, exactly. Although I have a feeling that it might be Twilight fans that end up being patient zero.
Really? Why?
Well, okay.
I have to hear this.
So let's say it's a successful virus, right? And they're carriers, right? Because doing it straight out, right, would be too obvious, not elegant enough, right? So somebody manufactures whatever. They somehow expose these, Twilight folks to the virus, okay? Aerosol, what have you. And then, they like start biting each other because they think it's like the sexy vampire thing to do,
Oh dear.
Make it all
Right?
sparkly.
right? And then then they transmit that to other people. And that's where it begins.
I think it's going to be
Yeah.
a convention.
Well,
Yeah.
That's
Could do
Twilight
that.
Convention.
Twilight Convention.
You want to mix the two together, go right ahead.
Horrible. That's a new form of con crud.
Now,
Oh.
we went to what's that nursing convention that we went to in LA?
The National Magnet Convention.
Okay.
Yeah.
So I was walking around there, and it's like me and how many thousands of women. And the whole time I'm just thinking, all of these people come from all of these different hospitals, and they've been exposed to all of these horrible different things, and now they're all standing in the same building. This is a terrible, terrible idea.
Oh,
Um,
yeah, that's a good point. I didn't even think about that.
So something like that, I don't know. That's
We are probably carriers to multiple diseases.
Yeah.
Yeah. That might be interesting. That might be a good idea, just to swab the noses of like 7,000 nurses to see like what we are holding.
Oh yeah, I'm sure you'd find
All
kinds
sorts of
interesting things.
all sorts of awesome things. Huh. Interesting. So then so you also have a wet lab. So
Mhm.
if you were to set up a lab to study the zombie virus or bacteria, well, probably not bacteria because virus is a little more difficult to deal with. Zombie virus.
Might
What?
have been some with bacteria. It just depends.
Okay, all right. We'll do maybe it's a prion. Ooh,
Yeah, there have been some with prions.
I love me some prions. That's the mad cow disease.
Yeah.
Mhm.
Oh yes, that is an elegant. Okay, sorry. So if you were to set up a wet lab, what would you physically need? Like in terms of equipment.
hmm Oh, well, we have incubators on hand, which you'd need to do either basic bacterial culture or grow cells and do tissue culture for viruses.
Sure.
Um, again, you probably want to do some molecular diagnostics. So we have thermocyclers and things like that, where we could sequence the bacteria, viruses, what have you, and determine if they're related to anything that we know of or if they're completely novel. What we don't have in my lab, so my lab is a BSL-2 facility.
Mhm.
So that just means that most of the stuff we do, we can do on the benchtop. Some of the stuff we do in the little, and we have hoods. But we don't have any extreme containment facilities, which is really where you'd want to do this type of work. Is a lab where you're using like the full body, kind of spacesuits type of things, or at least at the very least, a backpack respirator or something like that. Because
Wow.
working with MRSA, almost it seems pretty safe compared to working with a zombie virus. So
Oh man.
Yeah, you don't
Wait,
end up
so the
biting
step-
each other if you get
Yes.
infected.
So
Yes.
okay, so last week or 2 weeks ago, we had talked to someone who set up one of those DIY BSL-1
Mhm.
labs. Like it's a community lab, and they work with relatively harmless strains of like E. coli.
uh,
This week, you have a freaking awesome biosafety lab 2, biosafety level lab 2.
And if the
Right.
CDC
And
returns
it-
our calls, maybe we'll get what level
3,
are they?
4, 5, and,
Yeah.
and
well,
not,
it
not
goes up
even.
to 4. Yeah, 3 and 4 are the
That's
ones
what
where
they
you have to
tell
have the
you.
respirators and
That's what
and
they
and
tell you.
4 are the full body suits.
Okay, so 3 is the rest. Man, that's just freaking awesome. Okay, so the division between 2 and 3 is the backpack respirators.
Yeah, and you have to also have negative air pressure in 3 so that anything basically that's in the lab stays in the lab and doesn't get released to the outside.
Fascinating. We
And
might
you
just
have to also shower in and shower out of those.
really of 3
Yep.
as well.
Yep.
Wow. That is so cool. Like I am so like sparkly eyed right now. That is excellent. Um I
Gonna be
kinda
okay there,
I
Annie?
kind of want a BSL-3 lab.
Really? Why?
Maybe 4. Because like the suits with the freaking like helmet things
Mm-hmm.
and the
Yeah. And when I was in high school, that's what I wanted to do. I wanted to work with Ebola and the BSL-4 lab, but then
dude.
having kids and stuff kind of
hampers
that
it.
a little bit.
Sure.
Yeah.
And actually we've been hearing a lot more about Ebola lately.
Yeah, there've been a couple outbreaks in Uganda.
Yeah. I mean, is it the same strain as what was dealt with way back when?
I can't remember. So there's actually 3 filovirus outbreaks in Uganda. One was of Marburg, so
Okay,
kind of
yep.
the Ebola cousin. And I can't remember if one was Ebola Sudan and one was the new Bundabujo, I think. But it wasn't Zaire. Zaire is the nastiest one.
Okay. Man, because when I hear Ebola,
Yeah.
first I'm like, what? I thought that was gone. I thought we eradicated that.
No, still around.
Okay.
So I did have a question.
Mhm.
You mentioned growing cultures and sequencing. A couple weeks ago, we were talking about that as well, and it sounded like they're sort of getting more away from the growing culture side of things because you can only grow the things that eat the sugars and whatnot. So it makes more sense now that we can do sequencing to just do the sequencing so you can get a much more clear picture of what's actually going on in your sample.
Well,
Has that been
kind of.
affecting what you do there too?
So there are pros and cons. So with sequencing, you can, sequence everything that's there. The problem is, one, it takes a lot of bioinformatics, a lot of computer nerdery to kind of put it all together. And, because it's really kind of assembling a puzzle from all of your different DNA sequences. So with that, you know it's there, but you don't necessarily know what's causing the problem.
Yeah.
So if you have a novel virus or something, well, you might be able to find its sequence in there, but you still have to prove that it's actually the thing causing disease, right?
Mm-hmm.
And so the same with bacteria, you can sequence all of that. You can know you have, 500 different species of bacteria, but you can't really characterize them very well if you don't have the cultures. Like for us, we do a lot of molecular analysis on the MRSA we get. And in order to test for, all the different antibiotic resistances, for example, we have to actually have a culture of that. Or to do any, any tracing of maybe where that came from. We have information about its genome sequence and not just, usually when they do, they just look at one particular gene called the 16S RNA gene. So it tells you if that species is there or not. So it doesn't tell you, about the characteristics of that bacterium. So, for us, it really helps to have an actual physical isolate of it and not just saying, the DNA is there.
Right. Because you can't expose the DNA to an antibiotic and be like, oh, look, you have to see like the actual cell colony dying.
Right. Or if, if it's a new virus or something, you can't put just the DNA in an animal model and, see if it causes a zombie disease or something like that.
Zombie mouse.
Yes.
Those suckers will bite.
Absolutely.
Yeah. And actually, in general, because, you are co-director of, let's see, what is this amazing sounding? The Center for Emerging Infectious Diseases. Right. So you would be, like, on the front line when it came to a zombie apocalypse.
Maybe. If I wasn't, didn't gather up all my things and go hide in the woods.
Don't worry, we'll send you the coordinates to our bunker.
Okay, there you go.
But what does the center do, and what's your role at it? And
yeah, so we try to bring together the state and kind of in the surrounding states to investigate emerging infectious diseases. So we have a lot of collaborations with our state hygienic lab, who do all kinds of outbreak investigations of anything that pops up around the state. We work pretty closely with some of our veterinary colleagues since a lot of new diseases come from animals.
Sure.
So we just try to kind of form collaborations with people in kind of the general vicinity of Iowa to facilitate infectious disease research.
Super freaking cool.
Yeah, it's fun.
Super cool. Like, I am just enamored by infectious diseases. I mean, granted, I am on ZombieTech.
They're enamored with you too.
Yes, I guess
It's
they
mutual.
would be. So it is that time, sadly, where we ask the actual ZombieTech questions. This has all been a warm-up.
All right.
Just kidding. I mean, Okay. So, and we ask these questions to everybody.
All right.
So, first question. What 3 tools would you bring to a zombie apocalypse bunker?
3 tools for anything?
Yes.
Addie's no longer allowed to answer specific questions on this because she's too lenient. But yeah, basically, what do you think you would absolutely need?
Well, some kind of Swiss Army knife because I'm hoping somebody has some canned goods packed.
Yes.
Definitely a weapon, probably just bat, maybe an axe.
Okay.
Number 3. Boy,
So
I don't
a
know.
no reloader type here. Yeah.
I don't know. I'm not really good with guns or anything. So, yeah, I don't know. You got me stumped.
Number 3. Are you sure I can't give her hints? You sure? I have to. She's the infectious disease guru. Okay, look. Essentially, I'll tell you what Bradley brought last time. He brought like an MRI machine.
Oh, geez.
Yes. Okay. So as long as you can theoretically strap it onto your back, you can bring it.
Okay.
All right. So
Well,
that being said,
Mhm.
what would your third one be?
All right, so I'll bring my thermocycler so we can find out what is causing this particular zombie outbreak.
Freaking awesome! Because our last week's guest brought an incubator and a sequencer. So
awesome!
I
So
think
yeah, we can all collaborate together. That would be perfect.
Oh man, I am so excited! The zombie apocalypse better happen.
Oh boy.
And question number 2: what book would you bring to the zombie apocalypse bunker?
Oh, the Zombie Survival Guide.
Excellent. I wonder if I bought that. I think I made like one of those just random zombie book purchases. And I very rarely buy books. It's kind of strange, but yeah. I thought we'd have to. ZombieTech headquarters has to have one.
Absolutely.
Excellent. All right, I will accept that. And in terms of finding out like where all your information is and how people can like read stuff that you're working on, where can they find all that?
Sure. I'm pretty Googleable, but my blog is at scienceblogs.com/etiology, which I know is everyone's favorite word. AETIOLOGY. Yeah, my Twitter handle etiology, so I also post stuff there. The Center for Emerging Infectious Diseases does have a Facebook page, so we're on there as well. My blog has a Facebook page too, so kind of everywhere around the net.
Super cool. Now, I have a quick I know I pretty much said I was finished with my questions, but I actually kind of have one.
Don't you always?
Yes, I always do. So, you are one of very few people, I think, that I've read who takes your research to the blogs. How would you encourage other scientists who, it's a rat race for publications and for posters and all that. Like, how would you encourage them to take it to the blogosphere?
Screw that. It's a rat race for humankind, and if we keep locking what we know behind walls, we're dooming ourselves.
Well, and that's exactly it. I mean, for some of my papers, I'm sure that they get read by, 30 colleagues, maybe 50. Whereas my blog, I don't update it as regularly anymore, so the traffic is down. But at its peak, it would get 50 to 100,000 hits a month. So it's if you want an audience, if you actually want to talk about your science, or I don't always write about my own work. I write about about Ebola and plague and all these other fun topics. If you want people to who have an interest to find that stuff online and find good information and not the crap that's on so many of the websites. You have to be responsible to get that out there. You can't just sit back and wait for all of your colleagues to do it for you.
Right. I totally agree. And your blog is like hardcore. So, and because there's like words like Ebola and like SARS and actually autism. I think there's been quite a bit of talk about autism and vaccines, obviously. Thanks, Jenny McCarthy.
Yes.
And like, I mean, just deadly diseases and their names are all over her pages.
Absolutely.
Which is just, that's got to be good content. Exactly. It's got to be good content. So, okay. Excellent. I will put obviously links to all of that. And, yeah.
Here at ZombieTech, we have a good respect for PhDs that share what they know with the world. We like that.
Yes.
Oh, well, thanks.
Agreed.
All right. Well, that's it for us for this week. That's all the time we have. Thank you for joining us.
Absolutely. Thanks for having me.
It's been lovely. You guys can find the show every Thursday at ZombieTech.tv. There's a RSS feed there on the page if you want to have it automatically downloaded to your phone, iTunes, or whatever. We're living in the future. Or you could just push play on the webpage. It's really easy. We'll be back next week. See you guys later.
Bye.
Bye.